Have you ever been wondering about why some people are more likely to get a disease such as Alzheimer's while other people don't? One of the possible answers that lie behind these kind of questions is that people are genetically different. In the last few decades, geneticists spent tons of efforts trying to understand how genetic variations among human beings can cause people to develop different kinds of diseases. But still, a big part of that still remains unknown. Recently, more and more researchers started to think about the role of non-coding regions. They make up of 99% of the genome, but we used to think them as junk DNA. Now, thanks to the new generation sequencing, people can sequence every base of the human genome, including the non-coding regions. However, analyzing those enormous genomic datasets is very challenging. Therefore, people have been seeking to develop powerful tools to help us prioritize non-coding genetic variants. And that's what I study. Particularly, I'm interested in one type of genetic variation, single nucleotide variants, where a single nucleotide in the DNA sequence is altered. Current variant prioritization tools make use of this huge amount of genomic dataset and use a computational approach to derive a single score to prioritize genetic variants. However, most of these tools people have right now are not disease-specific. For instance, we are using the same tool for Alzheimer's and inflammatory bowel diseases. Therefore, my research goal is to explore approaches to develop tools that works for a specific disease. One project I'm working on right now is to improve on one of the current variant scores to derive a disease-specific variant score in more than 100 diseases. How can I do that? First, I compile a benchmarking dataset that contains known disease-associated and non-associated variants in more than 100 diseases. Next, I use a computational approach to generate disease-specific tools using the dataset we generated. We found that this disease-specific approach has a better performance than other current methods. In addition, we learned something interesting from our model. We can learn which cell types are relevant to a disease and how those diseases are similar to each other. Overall, my work suggests a disease-specific expertise in studying genetic variation. And by developing a disease-specific tools in the future will help geneticists to unravel the hidden mystery of our human genome.